Let's continue on with a basic review of the cell cycle. Cells reproduce by duplicating their DNA and cytoplasmic organelles, then dividing into two. This process spans all living organisms from unicellular bacteria to multicellular species. Cell division and duplication of DNA must be carried out in a faultless manner because the survival of the species depends on it. To this end, there are controls exerted during duplication of DNA and during cell division to prevent errors, identify errors if they do occur, and to correct those errors. DNA is the only macromolecule that undergoes repair to ensure the fidelity of the information it carries to the nucleotide sequence. The most conspicuous stage of the cell cycle is mitosis, the process of nuclear division, which is a prerequisite for cell division. There are four stages of the cell cycle as follows. G1, the interval between the end of mitosis and the beginning of DNA synthesis. S, which is the replication of DNA. G2, interval between the end of DNA synthesis and the beginning of mitosis. And M, mitosis. If errors in DNA are not corrected, this will impair genomic stability and serve as a major contributor to the development of the malignant phenotype. Damaged genes that regulate cell proliferation and DNA repair are amongst the principal targets that cause genetic damage that sends a cell down the path to malignant transformation. These genes include the following. 1. Proto-oncogenes that promote growth and that function normally in utero and at puberty. 2. Genes that halt progress through the cell cycle, the tumor suppressor genes. And 3. Genes that carry out repair of DNA. And 4. Genes that regulate cell death, so that cells whose DNA cannot be repaired are forced to undergo apoptosis. The cell cycle control system is regulated at specific checkpoints, the two most important of which are at the interface of G1/S and G2/M. If DNA damage has occurred by irradiation or chemical alteration, normal cells will be prevented from entering S phase and replicating the damaged DNA. Orderly progression through the four stages of the cell cycle is orchestrated by a set of protein kinase proteins called cyclin-dependent protein kinases, or CDK, which, like other protein kinases, phosphorylate selected proteins on serine and threonine residues, thereby activating the proteins. To control this awesome power of cell division, a second family of activating proteins called cyclins bind to CDK molecules, thus modulating the ability to phosphorylate target proteins. In short, the cyclic assembly, activation, and dismantling of the cyclin-CDK complex is at the heart of the cell cycle. The G1/transition point is also known as the restriction point, and by passing beyond this point, normal cells are driven toward DNA replication. It makes sense that some monitoring system must determine that the DNA is fit to be replicated in the S phase, because replication of damaged DNA can be a prelude to a malignant transformation. Further, there must be a sensor to determine that replicated DNA is fit to undergo mitosis. A key sensor at G1/S and G2/M is a transcription factor, p53, which has been called the guardian of the genome. p53, acting through two downstream proteins, inhibitor of kinase 4/alternative reading frame ARF, blocks the cell cycle by acting as a brake in the cell cycle when p53 detects damage in DNA. Acting at G1/S, cells with defective DNA will not be permitted to replicate that DNA. Acting at G2/M, p53 monitors whether or not DNA replication is complete and whether or not mitosis can commence. It is clear to see why nearly 50% of cancers harbor a mutation in the gene encoding for p53, thus neutralizing a powerful protector of the genomic integrity. Another protein of overriding importance in monitoring progress through the G1/S checkpoint is the retinoblastoma RB susceptibility protein, which normally prevents cells from replicating by forming a tight, inactive complex of E2F/DP1/RB that alters the 3D shape of chromatin, making it more compact and therefore unable to replicate. In a normal cell, cyclin D slash CDK phosphorylates RB, thus allowing replication to proceed. In a cell with damaged DNA, RB senses the damage, resists phosphorylation, and thereby inhibits DNA replication until the damage can be repaired or the cell is forced to undergo programmed cell death. If the gene for RB is damaged, um, as it is in many different cancers, the result is unrestrained cell division, one of the hallmarks of the malignant cell. And finally, for this video, we're going to talk about tumor suppressor genes. Tumor suppressor genes act as brakes on the cell cycle. Failure to stop replication of damaged DNA can lead to the malignant phenotype. These two genes, p53 and Rb, are the most important tumor suppressor genes that are altered in cancer. Insight into the role of Rb was gleaned from investigation of the rare retinoblastoma tumor of the eye, in which 40% of cases are inherited as a predisposition to develop the tumor. The explanation for how a tumor could develop, both on a genetic and sporadic basis, came from Knudson's two-hit hypothesis, which proposes that in inherited cases the first hit is inherited from one parent as an autosomal dominant trait and is therefore already present in each somatic cell. The second hit occurs when one retinal cell that will go on to establish a malignant clone. In contrast, sporadic cases require that a single cell suffers two hits to the Rb gene. It is noteworthy that patients with familial retinoblastoma are also at increased risk for osteosarcoma, implicating the Rb gene in the pathogenesis of that bone tumor. Inherited mutilation or loss of the p53 gene is associated Inherited mutation or loss of the p53 gene is associated with another inherited predisposition to cancer, the Li-Fraumeni syndrome. This heritable syndrome predisposes individuals to cancers involving the breast, adrenal glands, brain, and sarcomas. Because the normal state for autosomal alleles is to have two alleles which code for a functional protein, when both of the normal alleles are lost, this is termed loss of heterozygosity, or LOH. Other tumor suppressor genes like BRCA1 and BRCA2 in breast cancer are not only involved in controlling passage through the cell cycle, but also regulate programmed cell death.